Hello everyone, welcome to the new session of plant tissue culture. So this is the fourth part on this topic, synthetic seeds. So here we are going to study about synthetic seeds. Synthetic seeds are encapsulated somatic embryos. We have already studied about the somatic embryo and the somatic embryogenesis in the previous session, right? So after the formation of somatic embryo, that particular embryo get encapsulated. Okay, not only the somatic embryos, shoot buds, cell aggregates, or any other tissue that can be used for sowing as a seed or that possesses the ability to convert it into a plant under in vitro condition or ex vitro conditions, and that can retain its potential also after the storage. Means this particular synthetic seed is having an embryo inside it. So this is a chemical based encapsulation of somatic embryo, and it is having a capability that it can form a new plant under in vitro or ex vitro conditions. Okay, it is having the potential. So synthetic seeds are produced by encapsulating a plant propagule in a matrix which will allow it to grow into a plant. Okay, so this encapsulating material is a matrix like the seed, natural seed only, right? So encapsulation of the micro samples. What are these samples? These samples could be somatic embryo or the buds. Okay, so the plant propagule consists of shoot buds or the somatic embryos that have been growing aseptically in the tissue culture. Okay, encapsulation is a necessary to produce and to protect the synthetic seeds. So this somatic embryo, when get encapsulated, it is called a synthetic seed. Okay, so the encapsulation is done by the various types of hydrogels which are water soluble. So this hydrogel encapsulate the embryo, that is somatic embryo. This is the hydrogel, and it get dissolved when water is there. So it is water soluble. The gel has complexing agent which is used in varied concentrations. So here are some of the agent or the chemicals which used for the encapsulation of the synthetic seeds. Okay, so gelating agent. What are the gelating agent? Formation of gelating agent. What are the chemicals required? You can now here see the sodium alginate with gelatin, carrageenan, locust bean gum, and gallurite for the gelatin agent. And complexing agent, calcium salt, calcium chloride, potassium chloride, ammonium chloride, and lower temperature, right? So these are the requirement for the synthetic seed encapsulation. Application of the synthetic seed. Now after the formation of this synthetic seed, what is their application? Why we are forming that? Artificial seeds provide low price production. It's going to act as a distinctive delivery system. It play a task of reproductive structure in the embryo development. Artificial seed technology has evolved as another and probably economic technique for the mass propagation of the various plant varieties. So if we want any plant in a large quantity, okay, so then we can go for the artificial seed technology, right? So somatic embryo is very easy to form with the tissue culture technique, okay, with the formation of the callus. And when these embryos are formed, they get encapsulated with these synthetic material. The encapsulation form a synthetic seeds. Now these seeds can be delivered from one place to the another. Okay, and they are also having the reproductive structure in the embryo development. So if we want a mass propagation, okay, with the various plant varieties, then we can have this artificial seed technology. By the employment of artificial seed technology, species may be propagated. Different species can be propagated with the help of artificial seed technology. Artificial seed may be transported from one country to different while not any obligations from the quarantine department, right? Because the natural seeds we cannot take from one country to another because of the quarantine, right? So we do not want, actually the country do not want any type of pathogen, okay? Because the seeds do have some types of microbes or the pathogen in that. And if they are transported from one country to another, they can introduce to that country also. But artificial seeds do not have any contamination or the microbes or the pathogen inside that, right? So it is easy to be transported from one country to the another. And there is no obligation from the quarantine development as well. Direct inexperienced house and field delivery of the elite seeds, chosen genotypes, genetically built plants area unit doable. So we can directly deliver that seeds into the field. Artificial seed crop area unit sometimes straightforward to the handle attribute to the uniform genetic constituent. So uniformity in the genetic constituent while forming the synthetic seed, because they have been formed from the one callus. They have been from the somatic cells, right? So their genetic constituent is uniform. Cereals, fruits, healthful plants may be studied with the assistance of artificial seed development at any place in the world. Artificial seed area unit terribly little, therefore they are straightforward to handle. It is easy to handle also. Now, another topic we are going to study in this session is this formation of the hybrid, that is also called as cytoplasmic hybrid. What is that? Hybrid are the cells or the plants containing nucleus of one species and the cytoplasm of both the parental species. What is that? So here, let us see. If we are taking the cell A, the parent protoplasm with nucleus plus cytoplasm. So cytoplasm is also there and nucleus of this cell A is there. But what about the cell B? Cell B nucleus has been taken out. It has been taken out. The parent protoplasm without nucleus. So only the cytoplasm of cell B is taken and both the cells are fused in vitro condition. Now this hybrid, this particular cell is called hybrid with two parental cytoplasm, and that is why it got its name hybrid instead of hybrid, right? Because the cytoplasm of both the parental cell are there and the nucleus of just one parent is there. Okay, so that is why it got its name hybrid. So this can also be formed in another way, right? We are taking both the cell, suppose in the second way, cell A and cell B are taking two parent cell, both the nucleus are there, and when these two cells are fused, there comes a stage that is called as hybrid heterokaryon. The nucleus of both the cells are not fused. Cytoplasm is fused, but the nucleus is not fused, right? So we have taken out nucleus of cell A from this hybrid heterokaryon, and we are having just nucleus of C cell and the protoplasm of both A and C. So this is again we get the hybrid. So either we can adapt this form or this form for the formation of the cybrids, right? So these are generally produced during the protoplast fusion in variable frequencies. So actually this is the fusion of the protoplasm. Cybrid formation may result by the fusion of normal protoplast of one species with enucleated protoplast, elimination of the nucleus of one species from the normal heterokaryon, gradual elimination of the chromosomes of one species from a hybrid cell during the further meiotic division. So this is very, very important, formation of the cytoplasmic hybrid. Actually, there is a hybrid of just the cytoplasm. The nucleus of one cell is there. One parent cell is there. Okay, so that is why it is not called as hybrid cell. Instead of hybrid, it is called as cybrid. That means cytoplasmic hybrid. Plants or cells which containing nucleus of one species but cytoplasm from both the parental species. Okay, so this is the cybrids, and they are also called as cytoplasmic hybrids. That involves the fusion of two protoplasm, donor and recipient, in which the donor nuclear genome is eliminated while its plastome and or chondriome are merged with that of the recipient. That is the protoplasm. For production of cybrid, cell uses two parental cells but should be one complete cell, that is cytoplasm with the nucleus, and other cell without nucleus or the inactive nucleus. So this is very, very important, and this we have already discussed, the two way of formation of the cybrids. Next topic is the cryopreservation. This is a very important topic in the tissue culture technique. The process of storing or preserving the biological samples in extremely cold or sub-zero temperature in a deep freeze, commonly at minus 196 degrees centigrade, is termed as cryopreservation. Okay, so whatever the storing material or the biological sample is there, we keep it at a sub-zero temperature, that is around minus 196 degrees centigrade, and that is called as cryopreservation. The word cryopreservation is derived from the Greek word kryos, meaning icy cold or the frost. At this particular temperature, whatever the biological sample is there, it get preserved. At such low temperature, all the biological activities of the cell stop and the cell dies. Cryopreservation helps the cells to survive cooling to the extreme temperatures and again thawing them to the physiological conditions. So, in the process of cryopreservation, when the extreme temperatures are removed, they can get back to the again the normal physiological condition. The ice formation inside the cell breaks the cell membrane and causes cell death. The freezing rate and the composition of the freezing medium are the two factors that can prevent this intercellular freezing. Cryopreservation is the use of very low temperatures to preserve the cells and tissue that are structurally intact. In this process, biological materials including cells, tissue, organs, oocyte, spermatozoa, ovarian tissue, pre-implantation embryos and other prepared culture media are preserved in extremely cold temperature for extended period without affecting the cell's viability. So the cells are viable, and what are the cells we go for the cryopreservation? You can see here some of the examples. That is the tissue cell, oocyte, spermatozoa, ovarian tissue. Okay, pre-implantation embryo also. Dry ice and the liquid nitrogen are generally used in this method of preservation. So liquid nitrogen is used. At these sub-zero temperature, all the biological activities of the cell get ceased. Tissue and other biological materials cease or effectively stopped and are presumed to provide indefinite longevity to the cell. Okay, the complete procedure step involved in the preserving the obtained biological samples are as follows. Harvesting or the selection of material. First of all, we have to select the material that we want to go for cryopreservation. Few important criteria should be followed while selecting the biological material, such as volume, density, pH, morphology, and damage free. The tissue should be, or the cell should be, damage free. Addition of cryoprotectant. Cryoprotective agents such as glycerol, FBS, salts, sugar, glycols are added to the sample as it reduces the freezing point of the medium and also allows slower cooling rate, which reduces the risk of crystallization. So there is no formation of crystallization in the presence of these particular materials. So we keep cryoprotective agents like glycerol, FBS, salt, sugar, and glycols, okay, in the medium. Freezing. Different methods of freezing are applied in this method of cryopreservation to protect cell from the damage and the cell death by their exposure to the warm solutions of the cryoprotective agents. Storage in the liquid nitrogen. Now comes the storage of this particular medium. The cryopreserved samples are stored in extremely cold, that is -80 degrees centigrade, in the freezer for at least 5 to 24 hours before transferring it to the storage vessel. So earlier it has been kept to -80 degrees centigrade in a freezer for at least 5 to 24 hours. And then it is transferred to the storage vessels. Thawing. The process of warming the biological sample in order to control the rate of cooling and prevent the cell damage causing by the crystallization. So this is the process of warming that protect the cell from the crystallization or the sample from the crystallization. Cryopreservation is a long-term storage technique which is mainly used for preserving the biological material without decline or decaying the biological samples for an extended period of time, at least for several thousand years. Now you can see this, several thousand years you can keep the biological samples or the biological material by this method. This method of preservation is widely used in the different sectors, including the cryosurgery, molecular biology, ecology, food science, plant physiology, and the different medical applications. So this cryopreservation is not only used in the tissue culture technique, but also in the cryosurgery, molecular biology, ecology, food science, and plant physiology. Other applications of the cryopreservations are in the seed bank, gene bank, blood transfusion, in vitro fertilization, organ transplantation, artificial insemination, freezing of cell culture, storage of rare germplasm, conservation of biodiversity, conservation of endangered and the disease-free plant species. So we use cryopreservation technique in so many areas of the biology. So this is all about this session. Thank you very much.